Okay, so can everyone hear me? Thank you all for coming to this talk, and I'd like to thank Boumediene for the invitation to speak. So I want to begin with an apology. There's not going to be a whole lot of, actually, in fact, there will be no machine learning in my talk. There is a data-informed, data-driven aspect to it that is mostly human-learned. That said, this is kind of the first step in a long-term project, and we are, so we've been able to do this first step by just very kind of old-fashioned multi-scale modeling, but I think going forward, I certainly expect the machine learning techniques to be very useful for us, and I've learned a lot already from all the speakers so far this week, and okay, and so that said, I'm just gonna jump in, and this is joint work with Duosen Xiao, my former student, who's now at Courant, and Lai-Seng Yang. Okay, so that works. Okay, so this talk is about so the only neural networks in this talk are going to be the biological kind, and so mainly this is about cortical modeling, and so for most of you know about this, but so let me zip through this. So the cortex is the outermost layer of the brain, and it's kind of a feature that's unique to mammals, I guess, and it's the seat of a lot of high-level neural information processing. And it's okay. So there's some dimensions. If you flatten it, it's kind of a couple of square feet and a couple of millimeters thick. So it's pretty flat structure, and it's divided into layers, depending on how you count, 6-ish layers, but actually a lot more. And then vertically organized into columns of 0.5 millimeter squared, roughly. And about 10 to the 10th neurons, 10 to the 4th synapses per neuron. These are kind of rough numbers that you'll hear. The things that and when you look at actual neurons, they actually come in a huge variety of shapes and sizes. So these are a couple of drawings, anatomical drawings, and they're incredibly diverse in terms of morphology and physiology. And so modeling has been very useful in computational neuroscience in making sense of all this, in summarizing experimental data where we can, and also in elucidating dynamical mechanisms. So what does the brain actually do when it actually computes something? And obviously, there are a lot of challenges here, just as with any kind of complex system modeling. Data, there's a ton of it, but it's very limited modalities. So we have very partial observations, and I'll say more about that in a bit. And when you make a model, well, there are kind of lots of parameters, at least by kind of pre-machine learning standards. Compared to a modern neural network, obviously the number of parameters is pretty small. Nevertheless, there's still a lot of them, so you got to worry about all the usual problems of overfitting and so on. And in the end, also you want to be able to rationalize this parameter space. You like to explain what's going on, right? You actually want to do biology. Okay. And then multi-scale dynamics and a lot of other things. All right, so this talk is an effort to address this the parameter question. So this is the first step in a project, in a longer-term project, as I said, and we doing what we can to constrain model parameters from data, and that's both anatomical and electrophysiological data mostly, and also making sense of the parameter space structure. And you'll see it's all based on very everything I say is only going to be approximately true, and nevertheless, and mathematically, in some ways very simple, but useful for biological insights, I hope. Okay. All right. So the specific setting is we're going to be looking at an area of the brain called the primary visual cortex. And specifically the primate primary visual cortex. This is a part of the brain that's very, very well studied, and so there's a lot of data about it. And so it's a good place to start for an endeavor like this. And there's been a lot of experimental and also modeling advances in the last several years. And so we're going to build on that try to address some of these kinds of questions that I mentioned, and our approach is going to be based on coarse graining some of these realistic but rather expensive models, and while preserving some degree of biological interpretability. Okay, so that's the goal. Okay, so a quick high-level introduction to the visual pathway. So this is the early visual pathway, and so visual stimulus comes in through the eyes here, and the part, so there's actually quite a bit of processing. Oh, I don't know how to point this thing. Okay, can I do this the old-fashioned way? Can I do that? Okay. I don't know if people online can see. Anyway, stimulus comes in through the eyes, and there's actually quite a bit going on already in the retina, which I'll mostly ignore. So the parts of the visual pathway that I want to highlight in this talk are something called retinal ganglion cells, which are okay. So this is now not stepping. Do you know what's going on with this? I see. It's not moving to the next slide now. I made the mistake of putting it down, I guess. Can I hit the space? Oh, no. There's not a space bar I can hit. Okay. Okay, so I'll just say it. So retinal ganglion cells are cells attached to the retina. They're actually part of your brain. They're the first stop in one of the first stops in the visual pathway. And then there's a step called the lateral geniculate nucleus, which is hard to say, so I'll just say LGN. And then the visual cortex is actually back here in the back of your head, about a credit card size area. Okay. And so that's the structure, and this is the main thing that I want to talk about. Okay. And so the primary visual cortex, so the LGN is part of the thalamus, and the first cortical area that processes visual information is the primary visual cortex. Okay. Okay. Oh, there you go. Okay, so it boxes those things. Okay, so I want to talk about tell you a little bit more about some fundamental concepts in this area, and so it turns out that there's a kind of a one-to-one mapping between cells along the visual pathway and location on the retina. And so there's a concept of a receptive field, which is the locations on the retina to which a cell responds. Okay, so for example, the retinal ganglion cells come in two varieties, so-called on-center and off-center. I'll explain what that means. And so if you take one of these on-center RGCs, it has a spot on the retina that it likes. If you shine light elsewhere on the retina, it doesn't do anything. But if you shine a small spot of light in the middle of that receptive field, so this is a picture of that, then you get a response. And as you make that spot of light bigger, you get a bigger response. And curiously, if the spot of light got too big, then the response starts to taper off. And that's what this picture shows. As the size of the spot increases, the response decreases. The off-center cells are similar because they do the opposite. Okay, so they like to spike when, okay. Anyway, so there are these two varieties of cells. For the purpose of this talk, LGN cells have very similar receptive fields. Like all statements in biology, this is actually debatable. Okay, but for the purpose of what I'm going to talk about, we're going to assume that that's true. Okay, so V1 is much more interesting in terms of its receptive fields. V1 turns out to be, so the cells in V1 have receptive fields that are still localized, but they respond to edges. Okay. And so what that means is that if you take a cell in V1 and you pass an edge through the middle of its receptive field, if it's the right orientation, it'll be very happy and spike lots. But if you give it a orthogonal edge, it will not spike. And so V1 is a kind of an edge detector. And so here's the pictures from David Hubel's book. And so showing that if you fix a particular cell, as you pass edges of different orientations to the receptive field of that cell, you get more or less response depending on the angle that that bar makes with the preferred orientation of that cell. And I should say, I forgot to add this to the slide. If you poke around V1, what you will find is that the preferred orientations of cells vary essentially continuously. Okay, obviously, nothing's exactly continuous. In biology, everything is very, very noisy and jumpy. But you definitely, if you look at the data, you will see that there's a kind of a continuity. Okay. All right, so now let me tell you a little bit about the this model. So everything that I'm going to do is based on a class of realistic models developed by Logan Cheriker, Mike Halkan, Bob Shapley, and Lyson Young. So this is a team that's based at NYU, and over the last decade or so, they've worked really hard on this model. Okay, and so the building blocks of the models are neuron models. And so single neuron models, as many of you know, so neurons are electrical devices. They operate by these voltage-gated ion channels in the membrane. And so you always start with Kirchhoff's current law, okay? Which expresses the conservation of charge, okay, or current across the membrane. And then depending on what you want to do, different neuron models do different things. We're going to take the simplest one, what are called leak integram fire models, where so voltage is basically the only variable that we have. This works a little bit like an old-fashioned flashlight. So if the voltage fluctuates, and then when you charge up to a certain threshold, you get a spike, and then it resets. Okay? So it's kind of a capacitor. And this is a relatively simple, this is a very simple model, right? So dynamics, except for the spike and reset, is linear. Okay. So all the interesting dynamics in these systems come from the fact that when you couple these things together, the timing of these spikes can get extremely complicated. All right. So I need to tell you a little bit more. In these models, there are what are called conductances. So the conductances describe the opening and closing of these voltage-sensitive ion channels. And I guess so for the purpose of this talk, I'm just going to ignore the fact that there are all these different shapes and sizes of neurons. I'm just going to focus on the fact that there are neurons that are excitatory. So neurons who, when they spike, cause the downstream neurons to want to spike, and neurons that are inhibitory. So that when they spike, they cause the downstream neurons to not want to spike. And the way that happens at a physiological level is that these conductance, each neuron has E currents and I currents. So these biasing potentials, what are called reversal potentials, are set so that when this conductance, when the excitatory conductance increases, that will tend to want to pull the voltage up, and then this pulls the voltage down. Okay, so that's the basic biophysics of neurons. And I should say that, forgot to say this. So each time a spike arrives, the conductance changes transiently, and how that happens is described by functions that are usually just taken as given. And there are timescales associated with that. And then the structure of the network is encoded in this kind of coupling matrix. Okay. All right. So now let me tell you something about the coupling matrix. So in the Cheriker, in this NYU model, so we're modeling, or they are modeling just a single layer in the V1. Did I say that somewhere? Anyway, so they're modeling what's known as the layer 4C alpha. So this is a sublayer of a layer in the visual cortex. This is the input layer in the visual cortex, and then just a few hypercolumns across, okay, 3 by 3. Okay, and this is tens of thousands of cells, and so each of these cells in the in this layer 4 are going to get inputs from LGN. Remember, that's the previous stop in the visual pathway. And so based on anatomical data, on average, each cell gets about 5 on and 5 off inputs. Okay, so pretty sparse inputs actually. Okay, not as many as you might think. And also based on anatomical inputs, we know something about the rough statistics of how E cells and I cells are connected to each other within the cortex. And so connection probabilities decrease with distance. And so you can model that by just some kind of truncated Gaussian. That's nothing special about the Gaussian, except that kind of fits the shape. And what's more important is these length scales. And then so you just draw connections randomly. Okay, and then so for E, I, and IE. And you have different distributions. And that's basically the model. This is a map of the preferred orientations, which I'm not really going to make use of in this talk. So I'll leave that there. Okay. Okay. So doing okay on time. Okay. So here are some results that you can taken from one of their papers. And so what this is showing, this is what's called a raster plot. Time goes this way. And these are indices of cells. So each dot is a spike. Red is excitatory. Blue is inhibitory. And what you can see is that when you and these are cells. Okay, so these are cells with similar, I guess, preferred orientations. So if when you give it a stimulus, so an edge oriented in the way that it likes, you can see that the excitatory cells are spiking a lot, and the inhibitory cells are also spiking a lot because the role of the inhibitory cells is to regulate the dynamics. Okay, so when E cells fire, I cells fire. If you give it a orthogonal grading, a orthogonal edge, you'll see that they fire a lot less. And then up their background means no stimulus at all. The cells fire even less. Cells do tend to spontaneously fire, so there's always some activity. Okay? And so this model captures that behavior also. And okay, so all of this is just to illustrate the range of things that these guys can do. And in fact, in neuroscience, a lot of the modeling papers that you come across tend to be focused on specific phenomena, like one or two phenomena, and then you try to build a model. And this is because building these models and trying to fit all the available experimental data is actually very hard. And so people do what they can. V1, because we know so much about it, and because these guys work really, really hard on it, this model fits easily, depending on how you count, a dozen or more phenomena with just a single model. Okay? Which, if you think about it, is really what you want, right? You only have one brain, all the phenomena should be reproducible by one model. Okay? But that's easier said than done. Okay. So in this talk, I'm going to focus on the boring part of the dynamics, just the background. Okay, so no input, just the background spontaneous fluctuations. Even in the background state, the dynamics is actually already non-trivial because the E and the I currents have to balance. And if one thing starts to dominate, either activity all dies down or everything blows up. And it's actually extremely easy to get these things to fire in a kind of a highly synchronized fashion, much more synchronous than this. You can see these rhythms here. And real brains don't do that. They don't synchronize completely, except when you're undergoing some kind of seizure or something like that. Okay, and so so there's a wide range of correlated activity. Okay, so more specifics. So we're going to so we have 3 by 3 hypercolumns looking at single layer, tens of thousands of cells. There's a dozen or 2 dozen biological parameters depending on how you count. We're going to focus on 7 that we care about, actually exactly where 7 is not too important to this talk either. I should say just a little bit more about the model. So the E conductances actually is a sum of a bunch of terms. There's a term corresponding to input from LGN. Because I'm looking at background, these LGN cells are just firing spontaneously, and so a Poisson process is not a terrible model, and so that's what we do. And then there are the terms that correspond to L4 cells connected to each other. So that's kind of a lateral connectivity term. And then there are additional things which I haven't talked about. In the brain, there are all sorts of ambient modulatory inputs. And also there's feedback from other layers, even within the cortex, and those turn out to be necessary to include these in the model to get realistic behavior. So those terms are also in the model. Okay. Um, uh, how am I doing on time? 5 minutes, okay, that's okay, yeah. Okay, so I can get through all the interesting stuff in 5 minutes. So one of the things I want to highlight is that if you look at these spiking neural models in detail, one of the very striking things is that the I cells really track the E cells all the time. And so in neuroscience, there's this concept of a balanced state where the E and I currents should balance each other. There are lots of reasons for this. It makes the brain extremely sensitive to small perturbations. If you have a sensory input that upsets this balance just a little bit, the brain will react. Okay. Okay, so that's the main thing I want to say about that. And so this is the same picture again. This EI balance also makes the dynamics very interesting, I guess, is the best way to say it. And so, and in particular, this kind of rhythmic activity is something that very easily happens in one of these models. Okay. And sometimes that's the correct behavior in some physiological conditions, and sometimes you don't want that. Okay? But this is kind of a dynamical property, and these are emergent phenomena. So there's no periodic forcing being applied to this model. Okay, so now to the main point of the talk. So in the last few minutes, and so when you look at the parameter space of these models, there's a kind of a conundrum. On the one hand, if you perturb the parameters by a small amount, you very easily get responses that are unreasonable. In this talk, I'm going to be looking at just very basic statistics like the mean firing rates. And so for example, so this is a little table that we made. These are the parameters, and if you that in the model, and if you perturb some of the parameters by a couple of %, you very easily get firing rates that are just physiologically unreasonable. Nevertheless, biological modeling works are pretty robust. And so if these models have any kind of relation to reality, they really ought to be pretty robust also. And also this NYU team really could tune the models by hand, okay, with in not astronomical amount of time. Because in a reasonable amount of time, they're able to tune these models. So obviously the chunk of parameter space that correspond to realistic behavior is not vanishingly small. Okay? And so how do you account for all those? Those are a couple of questions we want to answer. And so here's the approach of what we actually did, what the part that I'm actually involved in, so we so the realistic model is too expensive to really use as a method, as a tool for mapping parameter space. And so we make a mean field reduction using ideas that are in spirit very close to equation-free modeling and heterogeneous multi-scale modeling techniques, for those of you who know about those things. And then as Nathan said in his talk, coordinates do matter. And so I want to explain how we can rationalize or explain some of the things that we see, at least at a very rough conceptual level. And then, as I said, the main thing that we will do is constrain the E and I rates. Okay, so all right. So the mean field part is actually I'm going to zip through because it's not. There's nothing. The ideas are actually pretty standard. And so you start with the integral and fire equations. You time average this. There's a long history of this in neuroscience. And then you get equations relating the firing rate to the mean voltages. You have to make the usual kind of physics-style mean field approximation where you ignore correlations between conductances and voltage. Okay, so this is an uncontrolled approximation. And then you say, well, I've got what's usually called a closure problem, right? I have a relationship between firing rate and voltage. I've got too many variables and not enough equations. And so what we do is so this is the point where classically in mathematical neuroscience, if your goal was to try to understand a specific phenomenon like pattern formation or something like that, you would just make up something that's qualitatively correct and then proceed with the analysis, and which is a very sensible thing to do. But here we're actually looking for some degree of quantitative agreement. And so what we did is we simulated small numbers of the single neurons. In this case, just one E and one I turns out to be enough, and then use that to augment these equations to get a closed model. Okay, so in the language of in the equation-free language, this is the mean field model is the macro model, and then you have the microscopic models. Okay, so let me get to the more interesting things. So here are the results. One of the results you can get. And so as I said, we were interested in 7 parameters, but we wanted to approach them somewhat hierarchically, right? There are ones that we care more about, ones we care less about. And it turns out the thing that's very useful to focus on are looking at these 2-dimensional cross sections where the axes are SEI and SIE. So I didn't explain the notation. The SEI, SIE are the coupling strings that govern how strongly the I cells inhibit the E cells, and SIE is how strongly the E cells drive the I cells. The reason you want to look at that is because they have a similar kind of effect. Okay. And so in this plot, the blue curves and the red curves are the level curves of the E and I firing rates. And what you will see is that they line up very closely with each other, and they have a roughly hyperbolic character. Okay. And you can wave your hands, because this is approximately correct, that the reason they have the same effect is because if E cells are driving I cells more, then you're going to get more inhibition in the system. And if I cells are inhibiting the E cells more, you're also going to get more inhibition. So at some level, they have they should have some kind of similar effect on the population dynamics. Of course, that's not exactly true, okay. But nevertheless, it gives you at least a heuristic explanation for why these curves look like hyperbolas. Okay, if you took this literally to be true, then you're going to get that's what you expect. And then more of this. And then the other thing that's curious is that the set of parameters that's compatible with the firing rates is roughly codimension 1. Now, if you think about it, if you have a space, if you have a submanifold that's defined by 2 functions that are, in some sense, independent, then you should expect generically codimension 2 level sets. And the reason that we get a neighborhood of a codimension 1 set again is because because of E and I track each other. Those 2 things are in some sense not that independent. And this also explains the why. It's in some ways the dynamics is pretty robust. If you vary 1 parameter at a time, then it's very easy to fall off the manifold. But if you get to vary 2 or more, then it's not that hard to stay on it. Okay. And I guess I've run out of time, so we can also explain some of the qualitative behavior in these pictures as you vary inhibition and excitation. So if you're interested in the biology, I'm happy to talk about this. Okay, so I'm going to end here. Those are the conclusions and various things. And can I have just 10 seconds? Okay, so a bit of shameless advertising. I'm myself and a bunch of my colleagues, we have a little NSF-funded research training group at Arizona. And we're looking for a couple of postdocs to start in the fall. The research interests run the gamut from including things that have been discussed here, physics-informed machine learning, applied to turbulence, power grid modeling, and so on. So if you are interested, come talk to me. Okay, so I'll stop there. Thank you.